Welcome to the Pharma Talks Biotechnology Careers playlist on research and development. In this series, we will review 30 technical interview questions covering molecular biology, genetic engineering, cell culture, analytical methods, and experimental design. Use each slide to practice a concise answer, then add an example from your own laboratory or research experience. Question 1. What is the central dogma of molecular biology? The central dogma describes the usual flow of genetic information from DNA to RNA and then to protein. DNA is transcribed into RNA, and messenger RNA is translated by ribosomes into a polypeptide. In an interview, I would also mention important exceptions, such as reverse transcription in retroviruses, while keeping the core principle clear. Question 2. What are the main differences between DNA and RNA? DNA and RNA differ in their sugar nitrogenous bases, structure, and biological roles. DNA contains deoxyribose and thymine and is usually a stable double helix. RNA contains ribose and uracil and is often single-stranded. Beyond carrying messages, RNA can regulate genes, form ribosomes, and even catalyze reactions. Question 3. How does PCR work? PCR amplifies a specific DNA sequence through repeated cycles. First, heat separates the DNA strands. Next, primers bind to complementary target regions. A thermostable polymerase then extends the primers using nucleotides. Repeating these steps produces exponential amplification. A strong answer should also name the essential reagents and explain why controls are necessary. Question 4. What is the difference between PCR, RT-PCR, and qPCR? Conventional PCR detects amplified DNA at the end of the reaction. RT-PCR adds a reverse transcription step, converting RNA into complementary DNA before amplification. Quantitative PCR or qPCR monitors fluorescence during each cycle to estimate starting material. Note that RT-PCR and qPCR are sometimes confused, so define your terminology clearly. Question 5. How would you design primers for a PCR experiment? Good primers are specific to the target, usually have similar melting temperatures, and avoid extreme GC content. I would check for hairpins, self-dimers, cross-dimers, and off-target matches. I would also choose an appropriate amplicon length, and for gene expression studies, place primers across exon boundaries when possible to reduce genomic DNA amplification. Question 6. What factors can cause nonspecific PCR amplification? Nonspecific amplification can result from a low annealing temperature, excessive magnesium, poor primer design, too much template, or too many cycles. I would first review the controls and gel pattern, then optimize one variable at a time. A temperature gradient, lower primer concentration, or hot start polymerase often improves specificity. Question 7. How do you evaluate RNA purity and integrity? RNA quality has two dimensions: purity and integrity. Spectrophotometric ratios can indicate protein, phenol, or salt contamination, but they do not prove that RNA is intact. Integrity should be assessed using a gel or automated electrophoresis system, such as a bioanalyzer. Throughout the workflow, careful RNase-free handling is essential. Question 8. What is gel electrophoresis and how does it separate DNA fragments? Gel electrophoresis separates DNA mainly by fragment size. Because DNA is negatively charged, it moves toward the positive electrode through a porous gel. Smaller fragments migrate more easily and therefore travel farther. A DNA ladder provides a size reference, while fluorescent or visible stains allow the resulting bands to be detected. Question 9. How does DNA sequencing work? DNA sequencing determines the nucleotide order in a DNA molecule. Different platforms use different detection principles, but the overall workflow includes template preparation, signal detection, base calling, quality assessment, and downstream analysis. In an interview, I would match the sequencing technology to the research question: required read length, throughput, accuracy, and budget. Question 10. What is the difference between Sanger sequencing and NGS? Sanger sequencing analyzes individual fragments using chain-terminating nucleotides and produces accurate, relatively long reads. Next-generation sequencing processes millions of fragments in parallel, enabling far greater throughput at lower cost per base. Sanger is ideal for small targets and validation, while NGS supports panels, genomes, and transcriptome-scale studies. Question 11. How does CRISPR-Cas9 genome editing work? CRISPR-Cas9 uses a guide RNA to bring the Cas9 nuclease to a complementary DNA sequence beside a PAM motif. Cas9 cuts both DNA strands. The cell then repairs the break through non-homologous end joining, often creating disruptive indels, or through homology-directed repair when a template is supplied for a precise edit. Question 12. How would you evaluate the off-target effects of CRISPR? I would evaluate off-target editing in stages. First, computational tools help select a guide with few similar genomic sites. Next, likely sites can be tested by targeted deep sequencing. For higher-risk applications, unbiased genome-wide methods may be appropriate. Results should be compared with untreated controls and interpreted alongside on-target editing efficiency. Question 13. What is a plasmid and what must an expression plasmid contain? Plasmid is a circular DNA vector that can replicate independently inside a host. An expression plasmid needs an origin of replication, a selectable marker, a promoter, and the inserted coding sequence. Depending on the host and application, it may also require a ribosome binding or Kozak sequence, terminator, tag enhancer, or secretion signal. Question 14. How would you confirm successful gene cloning? Successful cloning should be confirmed at multiple levels. Selection identifies cells likely to carry the plasmid, but it is not sufficient. Colony PCR or restriction digestion can confirm insert size and orientation. The final construct should be sequenced to verify the complete insert, junctions, reading frame, and absence of unwanted mutations. Question 15. What is the difference between transient and stable transfection? Transient transfection provides short-term expression because the introduced DNA usually remains episomal and is gradually lost. Stable transfection creates a population or clone that maintains the construct over time, commonly after selection. Transient expression is faster for screening, while stable cell lines are better for long-term, consistent experiments or production. Question 16. How do you select an appropriate cell line for an experiment? Cell line selection begins with biological relevance. I would consider tissue of origin, species, genotype, pathway activity, and whether the required phenotype is preserved. Practical factors include growth rate, culture requirements, transfection efficiency, and assay compatibility. Before use, the line should be authenticated, tested for mycoplasma, and tracked by passage number. Question 17. What are the essential requirements for mammalian cell culture? Mammalian cells require an appropriate nutrient medium, controlled temperature, gas conditions, humidity, and strict aseptic technique. Culture vessels or coatings must support attachment when needed. Seeding density and passage timing should be standardized because overconfluence or excessive passage can change cell behavior. Routine observation helps protect viability, morphology, and reproducibility. Question 18. How can cell culture contamination be detected? Contamination may appear as turbidity, unexpected particles, rapid pH change, abnormal morphology, or altered growth, but some contaminants are not visible. I would combine routine microscopy with sterility and mycoplasma testing. Any suspicious culture should be quarantined, and related reagents, incubators, and handling records should be investigated before restarting work. Question 19. What is mycoplasma and why is it a serious laboratory concern? Mycoplasmas are extremely small bacteria that often contaminate cell cultures without producing obvious cloudiness. They can change cellular metabolism, gene expression, growth, and treatment response, making experimental data unreliable. Prevention requires quarantine of new lines, good aseptic practice, routine testing, and appropriate decontamination or disposal of affected cultures. Question 20. How would you determine cell viability? Cell viability can be measured through membrane integrity, metabolism, or cellular ATP. Trypan blue is a simple dye-exclusion method, while Risazurin, MTT-type assays, and luminescent ATP assays support higher throughput measurements. The selected assay must fit the cell type and treatment, and results should include blank, untreated, and positive death controls. Question 21. What is flow cytometry and what information can it provide? Flow cytometry analyzes individual cells as they pass through lasers in a fluid stream. Forward and side scatter provide information related to cell size and internal complexity. Fluorescent labels reveal proteins, viability, DNA content, or functional states. Careful controls, compensation, and a logical gating strategy are essential for reliable interpretation. Question 22. How does Western blotting work? In Western blotting, proteins are first separated by gel electrophoresis and transferred to a membrane. The membrane is blocked, then incubated with a target-specific primary antibody and a labeled secondary antibody. Detection reveals a band at the expected molecular weight. Quantification requires suitable exposure and normalization to total protein or a validated loading control. Question 23. What controls should be included in a Western blot experiment? Positive control confirms that the antibody and detection system can identify the target, while a negative control reveals nonspecific signal. Loading controls or total protein measurements support comparison between lanes. Depending on the experiment, I might also include secondary-only knockout or peptide blocking controls to test antibody specificity and background. Question 24. What is ELISA and what are its major formats? ELISA uses enzyme-linked detection to measure an antigen or antibody. Direct and indirect formats are relatively simple, while sandwich ELISA offers strong specificity by capturing the analyte between two antibodies. Competitive assays are useful for small analytes or limited epitopes. Quantitative assays require standards, blanks, controls, and an appropriate calibration curve. Question 25. What is the difference between monoclonal and polyclonal antibodies? Monoclonal antibodies originate from one clone and recognize a single epitope, giving strong specificity and lot-to-lot consistency. Polyclonal antibodies contain multiple antibody populations that recognize several epitopes, which can increase sensitivity but also background and variability. The best choice depends on the target, assay format, and reproducibility requirements. Question 26. How would you design appropriate positive and negative controls? Positive control should reliably produce the expected effect and demonstrate that the system is working. A negative control should lack the relevant target or treatment and reveal baseline signal, contamination, or nonspecific activity. Controls must be processed in parallel with test samples. Where appropriate, vehicle, mock, and no-template controls isolate specific experimental variables. Question 27. What is the difference between technical and biological replicates? Technical replicates repeat a measurement on the same biological sample and help estimate assay precision. Biological replicates come from independent organisms, cultures, or experimental units and capture real biological variation. Technical replication cannot replace biological replication. The number of each should be justified by expected variability, study design, and statistical power. Question 28. How would you troubleshoot an experiment with inconsistent results? When results are inconsistent, I first confirm the pattern in raw data and controls. Then I review sample handling, reagent lots, timing, instrument settings, and protocol deviations. I prioritize likely failure points and change one factor at a time. Every troubleshooting step and outcome should be documented before repeating the full experiment. Question 29. How do you document experiments to ensure reproducibility? Reproducible documentation includes the exact protocol, sample identifiers, reagent suppliers and lots, equipment settings, dates, operators, and any deviations. Raw data and metadata should be preserved, while analysis scripts and processed outputs should be version controlled. A good record allows another qualified researcher to understand, repeat, and audit the work. Question 30. How would you design a complete experiment to test a biological hypothesis? A complete experiment begins with a testable hypothesis and a measurable prediction. I would select a biologically relevant model, define primary outcomes, and include positive, negative, and vehicle controls. Replication, randomization, blinding, sample size, and statistical analysis should be planned in advance. Finally, I would pilot critical steps and define acceptance criteria.